Okay, thank you, ma'am. So we will be starting at 11 as soon as most of the participants can join us. So
Okay.
welcome to our lecture series.
Okay, just let me test you. Everything is okay. Let
Yes,
me
ma'am.
upload my file and see.
Okay, ma'am.
Is it visible?
Yes, ma'am. It is perfectly visible.
And full screen, right?
No, it has not been full screen. Not yet. It is showing the PPT dialog only.
Now also it's not full screen.
No, ma'am. It is showing only the PowerPoint dialog, and in the left side, I can see all of the slides. It is not getting full screen.
Okay. Okay. Let me. Is it okay now?
It is now okay now. It is getting full screen.
Okay. And also changing.
Yes, ma'am. The slides are also changing.
Okay.
Right.
Okay. So we are ready. And
yes. We can just start in 1 or 2 minutes.
No problem.
Yes,
no
ma'am.
problem.
Actually, some of our participants are still joining, so we can just start at 11.
Okay, no problem.
Nivedita madam, I have joined.
Yes. As it is 11, we can just start the meeting. Welcome to all of you in the Science SETU Lecture Series 3, which is in collaboration with the National Institute of Biomedical Genomics. We have Ashwati ma'am here who will be delivering the Science SETU Lecture 3 for us. So before starting the event, I would like to request our principal sir to please share his words of wisdom for us. So please.
Yeah. Good morning to you all. Thank you, Nivedita madam, today's host. First of all, I would like to express my sincere gratitude and heartfelt thanks to Dr. Bhashwati Pandit, Associate Professor, National Biomedical Laboratory. We have a collaboration in the perspective of the Science Lecture Series. Despite her busy schedule, ma'am has agreed to share her vast experience. I can say the extensive experience in the field of tuberculosis. And we are very lucky, and students particularly. One honest request to my beloved students: please interact with such a eminent resource persons in the context of the your subject, that is tuberculosis, its origin, the independent day, then the research, everything. And we are very lucky enough in that sense. So without much delay, I can say one thing, that Science into Lecture Series opened up a new era, I can say, from a general degree college like Durgapur Government College, with such a renowned biotechnology laboratory, where ma'am and other colleagues, they are working in different dimension of the biomedical laboratory, etc. So we are very lucky enough, and I must express my sincere gratitude to Dr. Burima Gupta ma'am. She has motivated us to build up a relationship with through the science lecture series, I can say. And once again, my sincere gratitude to ma'am. And once again, my request, I can say, to all the audience, research scholar, faculty members, and please interact after completion of our lecture, and you will gather enormous experience or information from her. So, with these few words, I wish all the best of today's seminar. Thank you, ma'am.
Yeah, thank you, sir. It was nice to hear your words of wisdom. So we will now move up into the technical session. Let me introduce Bhashwati ma'am before that. So Dr. Bhashwati Pandit completed her BSc in 1993 and MSc in 1995 from University of Calcutta. She was awarded PhD by University of Calcutta in 2001, and subsequently she worked as a postdoctoral fellow from 2001 to 2005 in the Division of Endocrinology, Diabetes and Medical Genetics, Medical University of South Carolina, USA, and from 2005 to 2008 in the Center for Molecular Cardiology, Department of Genetics and Genomic Sciences, Mount Sinai School of Medicine, New York, USA. She then worked as assistant professor in the S.N. Pradhan Center for Neurosciences, University of Calcutta in 2008, and subsequently she joined the National Institute of Biomedical Genomics in 2010. With her very vast research experience, she has completed several major research projects, research articles in reputed international journals, monographs, reviews, and book chapters, and has also delivered several invited lectures in international conferences. She was awarded the Best Publication of the Year 2007 by Mount Sinai School of Medicine, New York, USA, and her research interest is to understand the genetic basis of disease that include infectious and complex disease. We are very honored to have you, ma'am, today with us. Ma'am will be speaking today on tuberculosis, a journey from ancient to modern times for the students and faculty members of our institution. So I would now like to request ma'am to please deliver the technical session. Ma'am, please.
Thank you, respected principal sir, and also Nivedita madam for giving this nice introduction. And I believe that this Science SETU lecture series is really useful for many multiple colleges. And in the past also we have been linked to some other colleges. So it's really great that this time we are linked to you, and hopefully the students and everybody will be benefited not only from your side but from our side as well, because we are also getting chance to interact with lot of students, lot of faculty members. So I think it's useful both ways. And I will also encourage the students, as I understood that not only biology but there are other stream students also. Therefore, I tried to make it very simple and very basic, so that it won't be it will be equally acceptable for the other stream students, but still, as a more focus on biology, there has to be certain biology. So the students, if you feel to interact or if you have any questions anytime or at the end, you can ask me. Okay, so let me go to the lecture. I hope you can see it, and it's in full screen mode.
Yes, ma'am, it is in full screen.
Okay, so tuberculosis, the name is no longer new to any of you. All of you have received the BCG vaccine immediately after your birth within few days, because tuberculosis is a health problem in a country like India, and therefore we are vaccinated. We need to protect ourselves. So today actually I will talk about some history of tuberculosis, not only as a disease, but what is the history of the pathogen and how it evolved with time, how old is the disease, and not only the evolve of the pathogen to accustom itself with modern times, but how we have also evolved. We meaning the host, not only the human being, but the animals also at time to time have evolved to accommodate the pathogen or have evolved multiple wise way to contain the pathogen as well. So let me see how much, it's a big history and big story, but let me see how in a small time I can convince you and tell you this story. So tuberculosis is an infectious disease, no doubt about that, all of you know. And possibly before COVID, we could have said that this is the second largest infectious disease. Now that COVID has spread all around the world and a lot of people are infected. So I would say other than this pandemic, the COVID pandemic, if you consider other infectious diseases, possibly after HIV infection, which has also spread around the world, tuberculosis is a major health burden. And WHO, the World Health Organization, it catalogs and gives a detailed report of every disease around the world, and they also have a separate report for tuberculosis. So according to this report, the most recent being in 2023, so they published the report till previous years, so that is the data of 2022. It suggests that around the world, 10.6 million new cases have evolved in last 1 year of 2022. And there are huge number of deaths also, about 1.5 million deaths. Now this number from 2020, 2021 and to 2022, it has increased from 10 million to 10.6. So we see that even though there is modern treatment or a vaccine, but still there is an increase in tuberculosis case around the world. So it's really a major concern. It's really a great threat and terror to humanity. Severe cases of tuberculosis infection is really fatal, and really people have to go infected patient have to go through painful life as well as painful treatment as well. And the disease is known or spread worldwide. There is less cases, there is more cases, but it has spread around the world. Some area have very high burden, and you will be surprised to see the most recent WHO report that India accounts the highest percentage of incidence. That has happened in 2022. So India accounts for about 27% infected cases around the world, followed by some other countries. So these 5 or 6 countries actually are always in the front line. Sometimes India is second, sometimes India is third, fourth, but sometimes first. But more or less, this centers around all these countries. Therefore, in this part of the world, it's really a major health burden, including India. So a lot of programs have been taken in India from the government. There are a lot of departments in the health science sector which takes care of newly infected cases, reporting of cases, providing them free medicine, etc. And once upon a time, it was thought that TB is a disease of poor people, and that is not true. A lot of rich people are also infected, and I would mention in the later part, like sometimes people are infected but they are unable to recognize that they are infected because sometimes symptoms are not there. Okay, but anyway, so let us proceed and see how the pathogen has evolved and what is the pathogen. So to consider infectious disease, we can group them in two groups, like one set called crowd disease, another set called older infection. From the term crowd, you can imagine that it requires or it is spread more among people where there is population density is more. Okay, so the pathogen needs to spread from one individual to other, and because of close proximity of people, there's a lot of people staying in one small place, or the density being high, it can spread. This is necessary for the survival of the pathogen because it needs a host to survive. Without host, it cannot survive. So it tries to spread in the population where more and more population is there. And this kind of disease is, or the spread is about say 10,000 years old, this group of disease. Here the pathogen is virulent, it quickly spreads and can cause the high virulent symptoms. And also it can transfer from human being to animal or animal to human being, which we call is zoonotic transfer. But some disease, some infectious disease, we see that it can slowly progress, or sometimes it can even be without symptoms, asymptomatic or latent. That is what we call latent infection. So what it does, it survives inside the host for long time and it adapts itself inside the host because it is not infecting many people, but in wherever the individual is infected, it has to survive. So the main focus or the intention of the pathogen is that it has to survive in the world. So it adopts different kind of strategies, and these kind of diseases were also seen in the pre-Neolithic tuberculosis lie. Is it a crowd disease or is it an older infection? So if you look into the few characteristics that I have mentioned, definitely we can call it as a crowd disease because we know that where there is like a larger number of people staying in small place, like in the industrial era where more and more people had to stay in a unhygienic and small crowded place, we know that there was incidence of TB or there was incidence of plague and so on. But at the same time, also we know that TB can also have a latent phase. That is where it is not showing any symptoms. So some pathogens or some clever pathogens have adopted in a nice way that you cannot classify them in a particular, or you cannot bound them in a very small characteristic features, but they can fit into any group because they want to survive. So TB, we can say that it's a crowd disease. We can also say that it's an older infection. It can blow up and show like full-blown disease. It can lead to full-blown disease. Also at the same time, it can also exist in latent form throughout your life. One may not know even whether they are infected with the pathogen because the pathogen is remaining like latent or dormant, and possibly it doesn't create any harm to that individual unless sometimes when the person is like immune compromised for some reason or for some other disease, so that time the pathogen might get activated and it might show up his face. Okay, so for tuberculosis we cannot classify in that way. It is clever pathogen, the mycobacterium, the causal pathogen, and it can spread in a high infectious mode as well as in a latent condition. So, what is the history and how old the civilization knows about TB? So, first, this person, Avicenna, was a philosopher as well as a physician in the country of Iran. He was present in the golden era of in the country of Iran, and he had first noticed that tuberculosis, the pulmonary tuberculosis, is spreading from one person to other, and he have also noticed that if the infected person is kept separate in a quarantine, so then the people can control the infection so that other people, it doesn't spread to the other people nearby. So he has actually documented many diseases in a very nice way, among which tuberculosis is one. So he confirmed that it's an infectious disease and you can limit the spread of infection if you quarantine the infected person. But the tuberculosis name was given much later, and the name was given because of the lung tubercle that is formed. The tuberculosis name came from that lung tubercle which is formed during the infection in lung. But still the causal pathogen was not identified, and only in 1882, all of you know, you have read in your course, microbiology course definitely, that Robert Koch, he identified the pathogen which is causal for tuberculosis, and for that he was awarded Nobel Prize. Okay, so actually after systematic investigation and record of Robert Koch's work, we know that how this pathogen is spreading the disease, and also he proposed the vaccines which is still in use, that is the BCG vaccine. And other than BCG vaccine, till now we do not have any other modern vaccine, though some modern medicines have been evolved because of other modern day problems of drug resistance and so on, but still that BCG vaccine is in use. So apart from this previous century, we know more about modern way to understand the disease, the pathogen. Very ancient time also, in China, in India, in Egypt, there was evidence of this disease. So about say 2700 BC around, and even little later in India, we see evidence. It's written in our older literature that there is some disease, some infectious disease, which is nothing but the tuberculosis. Even in older Greece history also we see the evidence of this disease in Bible. And as I said, like in Egyptian mummies, lot of mummies had indication of TB infection. There was evidence from the bone deformities or lung deformities, and even I would say, how is it because of tuberculosis or something, how it was known? It was known using this advanced genomic techniques to prove and to confirm that yes, they were infected with Mycobacterium tuberculosis. And there is lot of changes of the pathogen. The pathogen has evolved. So the modern MTB or the Mycobacterium species that we see, so one of the oldest evidence was found in a mother and child who were submerged in the Mediterranean Sea. Besides that, in other countries, other part of the world, like say in South America, even in North America, in human or in among animals also, different animals, the molecular studies have identified presence of Mycobacterium infection. Now, there is not only one Mycobacterium species, there is many. Some can infect the animals, some can infect human being. So let us have a look into that. Okay, so even in the clay tablets, etc., which are like the mark and the evidence of historical events, there is an evidence that showing in the clay tablet that patient is coughing blood. Okay. Now this disease, in India we have a different name besides tuberculosis. You know that we call it like in Bengali khoi rog, or in Hindi like shai. So all this, even the same meaning was used for other countries like in Greek and so on, which means actually consumption and wasting. So what happens is this in this disease is there is wasting of muscle, there is wasting of your other systems gradually, and therefore like blood comes out of sputum, and there is severe dysfunction, and gradually like all the organs, if it's even though in pulmonary it is the pathogens are residing in the lung, but eventually it is like affecting other organs and survival of the person. So we see that a similar symptom prevailed even in the ancient tuberculosis patient around the world, and that has been documented in several historical evidences. So the modern tuberculosis, we call it, it is caused by inhuman Mycobacterium tuberculosis, but it's a group of pathogen. It's called Mycobacterium tuberculosis complex. Okay. So this complex actually consists of multiple mycobacteriums. Some of them can infect humans, Some of them are uniquely infecting human to cause tuberculosis, and some of them are also infecting other animals, cattle, and so on. And also there is possibility of cross infection as well. Okay, so what kind of pathogens are there? They are obligate pathogens, and they can cause similar symptoms in bovines, in other cattle, and also in even like some other wild animals as well. Some of this mycobacterium species is present or can infect like fish, like birds, and so on. So let us see how and which species are like infecting humans and which are infecting other animals. The Mycobacterium tuberculosis or Mycobacterium is a gram-positive actinomycetes group of bacteria. So the ancestor species of this group, they were found in soil and in other environment niches, for example, water bodies, for example, among some other rocks and so on. Okay, and at that time, the bacteria was like from this group of ancient bacteria, some bacteria evolved, leading to formation of tubercular bacilli, which was like smooth in nature. Okay, that may not be a pathogenic Mycobacterium tuberculosis that we see nowadays. Again, with time, when they evolved, some new species came out of this, and they consist of, and possibly it happened in Africa, why I will tell you later, and possibly this smooth tubercular bacilli, or sometimes it is called STB, this gave rise to other species like Mycobacterium cannati, africanum, and so on. Okay, and from there, finally, there was further evolved virulent pathogen, the Mycobacterium tuberculosis, which caused tuberculosis in human beings as well. Okay, so besides tuberculosis, mycobacterial pathogen can also cause other diseases. So as I said, it is caused by the complex group of mycobacterium. Here are some examples. Another group of mycobacterium cause leprosy, which is called Mycobacterium leprae. Okay. Beside that, also there is group of mycobacterium which we call non-tubercular mycobacterium. They do not cause tuberculosis in animals or human, but they belong to the mycobacterial group, and sometimes they cause like skin disease and some other kind of infections, and like Mycobacterium avium complex or Mycobacterium um, marinum. These are some of these examples of this non-tubercular mycobacteria. So these symptoms resembles tuberculosis. They are tuberculosis-like, but they are actually not tuberculosis. So grossly, we can group the mycobacterial disease into three groups like this. So one set of pathogen cause tuberculosis, one set cause leprosy, and the other non-tubercular mycobacteria can infect with mild symptoms. But again, if some individual is immunocompromised, these mild symptoms can be carried over for a long time, leading to a chronic infection. Okay, so these are basically different kind of mycobacterial infection that we see. Now the question comes, what genomics can do to understand tuberculosis or the mycobacterium? Okay, so in modern times, using these genomic tools, we have come across lot of new ideas, lot of way to define and design new treatment strategies. And before treatment strategies to design, we need to know the pathogen in great details. Because as I said, the pathogen is very clever, and we need to know as a human being how we can still tackle the pathogen. So here genomics has helped us a lot. First thing before going into genomics, let me ask whether all of you have an idea about what is called genome and what is genomics. You may not have, but let me tell you. So, what we mean by genome? So, you know that all organism, you definitely have read biology in your school. So, all organism have certain genetic material in their cell, or if it is a multicellular organism, in every cell, right? So, this genetic material carries the information of life for that organism. And this genetic material can be transmitted to the next generation, or even when it's cell division, cell is growing from 1 to 2, 2 to 4, this genetic material also multiplies, so that each cell of all organism has the complete set of genetic material. So this complete set of genetic material is called the genome of that organism. It could be a virus, it could be a bacteria, it could be a plant or a higher animal. Everybody has their genome. But the genome content might differ. The composition or the basic building blocks does not differ. They are always like the four letters ATGC, the adenine, guanine, cytosine, and thymine. These are the four building blocks of the DNA, which doesn't differ, but the proportion or the composition or the arrangement of the back, all these four components differ. So what genomics has enabled us? Genomics has enabled us to study the genome in a multi dimension scale. Okay, so at a time we are analyzing the whole genome instead of looking into bits and pieces of that, which was done earlier. And all this is possible because of these high throughput techniques. So not only biologists, but here I can say students who are from different backgrounds, maybe physics, maybe mathematics, maybe computer science, you can all contribute to this development of this high throughput assay and analysis to extract all possible information from life. Okay, so the genome of tuberculosis was also analyzed, and now we know that what are the each and every base present in the mycobacterium genome. Okay, so here is a dialogue of a very famous scientist who worked in the area of tuberculosis. So once the genome of the tuberculosis was known, he mentioned that so one by one, people were developing techniques, and from very simple organism, we came to know the sequence, and we finally moved to human being, which has a very complex genomic organization and very large set of DNA. So when mycobacterium DNA, the sequence was done, he mentioned that this history, together with the clues to conquer tubercle bacillus, is written in its genome. So what he meant is that there is lot of information in this genome, but we have to know how to read it. Okay. So once all communities, meaning all the scientific communities from different streams come together and help everybody to know how to read it and what is written here, like kind of deciphering the information in the present in the genome. So that gives, or the sequence information actually gives us the knowledge about all potential drug target and of every antigen we may wish to include in a vaccine. So once we know about the whole genome or the genes present, we can design vaccine against the pathogen more and more. Okay. So vaccine designed against one gene may not be effective, but as we know more and more, this will be a very useful information to conquer the pathogen. So once the genome sequence of this mycobacterium was known, now let us see what we can do with that information. So roughly the mycobacterium tuberculosis had 4,000 genes, and we know some composition about the genome and some regulatory feature like it is not it's a variable, there is lot of features in the genome. Some genes are like constant, conserved across all organisms, some are variable. Therefore, this genome sequence and also the comparison with other mycobacterium or other species actually helps us to identify the information or the blueprint of information which is residing in the genome. Okay, let us see how genomics has helped. So, originally, we came using this genomic information, we could identify the original, the smooth tubercle bacteria. From there, the genomic studies has enabled us to understand how this has evolved to a professional pathogen, the Mycobacterium tuberculosis, causing the tuberculosis in the modern days. But this is not the end. Okay. Further, what is the problem we are facing nowadays? We in 18 after Robert Koch discovered the pathogen and vaccine, we thought we could conquer the disease, but that's not the case. So modern day problem is the pathogen has developed drug resistance. Okay, so it is clever. It is also trying to survive. So whatever drug we are giving to them, it is becoming resistant, it is still surviving. Also, not only it is restricting itself into a one kind of host, it is going to multiple species so that it can, if it not cannot, if we treat human beings, but it can survive in some other species. At the same time, also with change of this pathogen, the host immune system is also changing. Very long back, there was a less virulent or non-virulent strain. More and more virulent strains are coming. So our immune system is also changing and making itself more effective to conquer the disease. Now, how genomics has helped us to understand this pathway? Let us see. Um, we thought, or earlier notion was that in Africa, there was no tuberculosis. Okay. And to Africa, the tuberculosis came with Europeans. But in modern time, when we had analyzed the tuberculosis genome isolated from Africa and did the phylogenetic study. Phylogenetic study means you do the sequencing from the sequence of multiple pathogens, I mean, multiple tuberculosis. Say you analyze 500 multiple tuberculosis, mycobacterium tuberculosis, which has been isolated from patient. You do their genomic analysis, you compare the sequence with high throughput computational tools, and you define a phylogenetic tree. Okay, meaning some species will be close by depending on the information of their genomic contents. Some species might be little distant and so on. So this is like the basis of phylogenetic tree construction. So where from one species there is deviation with, or they try to deviate from each other by change of their sequence or change of the bases in their genome. So that genomic information helps to construct a phylogenetic tree, and it proves that the country of Africa or the continent of Africa was free of tuberculosis disease is not true. What happened is that on phylogenetic analysis and genomic analysis, people have found that the ancient species of Mycobacterium also exist in Africa now, which suggests that yes, Mycobacterium or the tuberculosis disease existed in Africa with a particular strain, but when with civilization and invasion of European and spread of European population to other continents for trade or for certain other reason, the Europeans carried the new strain from Europe to Africa. And therefore, nowadays in Africa, we see all kind of mycobacterial strains. Now, around the world with genomic analysis of mycobacterium, we can group them in seven groups. And all these seven groups are found in Africa. Okay, so what happened is when the Europeans came, they carried the new strain as well as the indigenous strain which was present in Africa. That also, in order to survive, that also acquired more and more fitness, because as you know that more you are fit, you will survive. Okay, so the transmission during this evolution or even earlier at some point of time, also the mycobacterium was transmitted to animals and animals to human. We had a notion that we got this tuberculosis disease from animals is not true. Okay, but current day, if we consider what is the transmission cycle of mycobacterium in nature, so we see that it can come from contaminated water, it can go to animals, it can go to human, and again from human it can go to back to the natural resources, or it can from human go back to the domesticated animals with which we are more close by, and so on. Okay, so it's always possible in many ways that the disease is spreading, but yes, the earlier evidence suggests that it has originated from a earlier species which was originally residing in nature in different natural resources. Okay, so genomics has also enabled us to break another concept that the tuberculosis is of animal origin and we got it from animal. No, the current day genomics based phylogenetic studies suggest that it existed in human, and at some point of time from human it moved to animals. Okay, so for some other disease the other way also it can happen, and the tuberculosis causing mycobacterium doesn't have any animal reservoir. It is not like, as you know, that during COVID, we know that it came from an animal resource entirely. But for MTB, it is not always true that it has come only from animals, but human beings are also for a long time harboring this pathogen. So I also mentioned earlier that from archaeological evidence, we know it's an old disease and where the mycobacterial species resided, and the sequencing, the genomic analysis of the DNA or samples that has been extracted from these archaeological samples also gives us more confidence. With genomic analysis, we are more confident to identify the species and so on. Okay, so this STB or the smooth tubercular bacilli, probably it had some clone of this one in Africa had given rise to the other species of mycobacterium, like Africanum and so on, and possibly all these complex or this species containing within this mycobacterium tuberculosis species migrated out of Africa with human. We know that human being, it is thought that humans or the modern humans originated in Africa, and from Africa they migrated to several continents, to Europe, to India, and so on. So the human beings along with them carried the mycobacterium, which may not be the current mycobacterium tuberculosis, but other strains as well. They have been carried out by the human. And on analysis of this mycobacterium around the world, as I was mentioning, that we can separate or group them into these seven lineage 1, 2, 3, 4, 5, 6, and 7 groups. And you can see that all of them are available or seen in Africa, lineage 6, lineage 5, lineage 7. Some of these are also present, lineage 1, 2, 3 is also present. Okay, and if you look into the evolutionary history which has been derived from this genomic analysis, so from these ancient species, first the non-tubercular mycobacterium had arisen, and from a certain point of time, so many billions ago, one of that evolved to make mycobacterium cannati, which had originated in Africa, and also during some with time, with age, the different other species has evolved, and at certain point of time, again from the human infecting mycobacterium, the Mycobacterium bovis has arrived, and also the other mycobacterium that can infect other organisms. So genomic studies again proves that it has not originated only, we did not, I mean human being did not get it from animals only, but at certain time point from human it also went to the animals. And here is the different lineages that I was talking about. So from Africa, as human population migrated in different region, these species or the mycobacterium also moved along with human. But definitely they have changed. They have also evolved. And what kind of changes? Let us see. So this is again earlier picture, like earlier migration from Africa. You can see one route has come to India, one has gone to Southeast Asia, then Europe and other places of of Africa. And then in modern times, when humans started spreading all around the world, like people looking for new world, people going for trade, and in modern days, like you can go anywhere. So that has again spread all over the world. So if you look into the timeline, uh, long back, as I was telling, the at very older times, we can call that they are present in the hominids, the much earlier form of modern humans. And at certain point of time from when it evolved, Mycobacterium tuberculosis came, and that along with migration of human population went to all of these countries. And from the literature of these ancient countries, we see that there was evidence of this disease. And as population expansion happened, the pathogen also moved all around the world. Here, the 7 lineages are shown how they are distributed along the world. So you see that East Asia, the blue color, the pathogen which is prevalent in this area, and the other color codes are here. And this is what is called the phylogenetic tree. So based on your genomic information, you can compare the different species, the sequence that has been isolated around the world, and you can construct the phylogenetic tree, where you see that this Mycobacterium caneti is one of the ancient Mycobacterium. Okay, so how did during this course of transfer or migration of modern human from different kind of these strains or the species of Mycobacterium has evolved? So the ancient one, the smooth bacillus, it can or it had accepted gene by horizontal gene transfer. So what is horizontal gene transfer? Meaning from one bacteria to other bacteria, the gene can get transferred. We know that plasmids carry certain drug-resistant genes and so on. So that is possible. Okay, but for Mycobacterium, the possibility of horizontal gene transfer is very rare in the modern Mycobacterium species. What has happened in the modern Mycobacterium is other events, complex event like recombination. Recombination means there is exchange of genetic material within the genome from one place to other. In higher organism, it can happen from one chromosome to other. There could be insertion, meaning new base can get inserted. There could be deletion, so you are deleting certain portion of the genome. And there could be single nucleotide changes. Okay, and then with these changes, when a new clone evolves, that clone gets certain advantage, selection advantage, survival advantage, so that clone then expands. So currently in the Mycobacterium genome, there are, if you consider other than insertion or deletion, if you just consider one nucleotide change, so there are about 20,000 such changes. And based on these changes across the 7 lineages, we can group them into those 7 groups. Okay, and also not that still it is constant, still it is evolving. If you analyze multiple patients, you might find still one or few more changes. Sometimes those are not lethal, they help in survival or giving better survival advantage to the pathogen. Okay, now coming to drug resistance, which is a modern day problem. So what happens? The pathogen has certain genes which is targeted by the drug that we give. So rifampicin is one of the drug that is used to treat mycobacterium infection, and what this pathogen does is it makes the target gene mutated, so that now the rifampicin cannot act on this gene or cannot block. Basically, it actually blocks the uh, transcription. Okay. There are other genes and other drugs used like ethambutol. So the EMB gene is mutated that confers ethambutol resistance, and there are some other genes as well. Now, simple drugs which were used as first line, then the scientific community have researched and developed some second line of drugs, but again the pathogen has become resistant to this second line of drugs, leading to multiple drug resistant pathogens. So multiple drug resistant people of multiple drug resistance is really a problem, and people are constantly developing new drugs to combat this pathogen. So here is a summary of how in modern times we have genomics have helped us to understand the pathogen. So first, as a landmark, as I said, that the complete genome sequence of MTB was known, and then not only the genome sequence, the different mutations were identified and expression of these mutant genes were cataloged. Now it was done around many countries in the world, so we have a information on the global population of the pathogen. Not only that, we also focused on the host. So in host, if we consider the human being for the Mycobacterium tuberculosis, so how the host behaves. So human genome has also been analyzed to understand how the host react towards the infection. And here also there has been evolution of multiple techniques. And here the complex human genome has been used to analyze, to identify or to know how the human genome is reacting towards this infection, and analyzing those human reactions or the reactions of the immune system, people have targeted host-directed therapies. Okay, so thereby genomic tools or genomics has enabled us not to understand the pathogen but also to understand the host. We also focus recently, India has started understanding the Mycobacterium species around the country. It's a nationwide study that has been undertaken, and with sequencing of a plan of like 30,000 Mycobacterium genome will be sequenced, and the aim or the goal of this study is to understand all the strain information prevalent in India, the lineages, the drug resistant pattern, and also the mutations. Okay, so that will help us to understand whether there are some new mutations prevalent in the mycobacterium within this country, so that we can design and develop new treatment regimes or new drugs. Now, as I mentioned or started talking about host, let us see, like if you consider a disease strand at one of each of these vertices, if I consider host at one, the pathogen in another one, environment in another one. So environment also plays a important role in manifestation of the disease. Okay, so this complex interaction between the host, the pathogen, the environment is leading, and the complex interaction between them is leading to the disease, of which host pathogen interaction is of immense importance to understand the disease. And sometimes the pathogen, actually the mycobacterium, can interact with other mycobacterium or other microbes rather. We know that HIV is the viral, people who are infected with HIV, they are immune compromised and they have high chance to get tuberculosis disease also. So when an individual is infected with both the pathogens, it's rather a more complex situation. So an infectious disease is actually a complex interaction of all three of these points, and effect of environment is really very important, but it's very difficult to dissect the exact role of environment. Okay, so in say last century, we you know that when people were infected with tuberculosis, they were taken to sanatorium, they were given good food, they were kept in well ventilated, well sunlit rooms, or even like outside in open sunlight. So how did that cure the patient? So here comes the role of environment. Good food, good nutrition. So you are boosting your immune system to combat the disease. At the same time, when you are in a good environment, meaning a well ventilated and well sunlit room, the sunlight it makes vitamin D in our system, right? And it has been proven that vitamin D induces a lot of antimicrobial peptides in our system that helps in killing the pathogen or combating the pathogen. Therefore, not only the host pathogen but environment also has important role. So, talking about few more words about the host. So, when the pathogen infects us, it goes to the lung, and actually the mycobacterium here shown in blue color is the mycobacterium. So, it surrounds itself with multiple immune cells of ours. Okay. So, why does it do that? The immune cells of ours comes here to control the infection so that they are limited within this small place. Also, the pathogen also, when it confines between these immune cells, it also protects from outside other cells. So it is both way good for the bacteria because the bacteria is confined within this small space and protecting itself. At the same time, the host, the immune cells, are protecting the host by limiting the spread of the infection from this side. And there is an aggregation of different kind of immune cells. These are called granulomas. Okay, so the host makes granuloma to limit the spread of the infection. And whenever any of us are exposed to infection, not that always we will develop the disease. Okay, so don't worry that whenever you are exposed, you will develop an infection. No, that is not the case. So most of us, like about 70% of the people, when exposed, they are able to clear the infection. Your immune system is so strong, so alert that you will be able to clear the infection. Only 30% of the people or the host can harbor the pathogen. Now 30% also doesn't develop the disease. Only 5 to 10%, if somebody's immunity is weak, they might develop the disease, and rest 90% of this 30% may able to harm without showing any symptom, that what I talked in the beginning, like latent condition, they might harbor the pathogen but there is no symptom. Okay, so when somebody is infected or exposed, only like 5 to 10% of people can develop the disease. Now, people with persistent or latent infection, they can go lifelong like that only if there is any reactivation of the pathogen under certain immune compromised condition, then only this individual can show up the disease. But yes, this people who are in latent condition they serve as the reservoir for the pathogen. So it's really a major obstacle to control the disease because you do not know who harbors the pathogen without showing any symptoms. So people are trying to understand from indirect studies like say gene expression of the host to understand whether they have got any infection or not. So if you consider with the iceberg, compare with the iceberg, so what we see on the tip is only a small fraction where the tuberculosis disease show up as a disease, but there is a huge fraction where the disease in a latent condition, and this has to be targeted to understand more and more and how to conquer the disease. So in terms of the host pathogen interaction, multiple factors from the pathogen and multiple factors from the host actually contribute. So the pathogen load could be high, there could be a more virulent strain, the pathogen might express more virulent genes, whereas the host tries to combat the pathogen through secreting many chemicals called cytokines. It tries to kill the pathogen with lot of cells that engulf the pathogen. And also, the most recently, we know that the contribution of genome is large to contribute or to control the disease. So, host genome has a lot of contribution to combat the disease. So, in the during the host pathogen interaction, so multiple factors come into play, and balance between them actually decides who will win. Whether the pathogen will win to spread the disease or the host will win to control the disease. Okay, so this host pathogen interaction is a very important aspect and the balancing aspect for the spread and establishment of the disease. So therefore, host gene plays a very important role, and in some cases, the familial aggregation, we see that certain disease prevalent in a family, even infectious disease. So that also led to a curiosity of scientists that why such happens. Is there any role of the gene in harboring such infectious disease in the family members? Yes. So there are evidence that one set of Mycobacterium disease is prevalent among a family members, multiple family members, and the cause for this disease to spread in a family is that they harbor certain mutation in the immune related genes. Okay, and that make them prone to infection with the mycobacterium. Besides that, people are trying to understand the gene expression, as I mentioned, of the host as well as the pathogen to understand what is the response of the host as well as the pathogen to understand the disease. Another kind of study is done where twins are compared to understand the genomics of the disease because twins share a lot of genetic information among them, and that gives us clue to understand many genetic component of multiple disease, not tuberculosis only, but any other disease. And grossly, what we can do to identify more and more host contribution of host genome or to identify more genes is that we carry out a comparative study of the genome of case as well as control, maybe in small scale, maybe in large scale, and build and test our hypothesis. Okay, so multiple host genome analysis can also give us clue how and what is the contribution of the host in manifestation and establishment of the tuberculosis disease. So we also in our research also focus on the host part besides the pathogen, as I mentioned earlier, and we carry out a comparative study of the genome of the tuberculosis patient and some controls who live within the same household of the patient, a family member, but never shows up symptom. That means this individual has certain protective factors and this individual is susceptible. Okay, so if we compare the genome using this high throughput genomic assay and tools, we can pinpoint to certain points in their genome which could be thought to make them protective or make them susceptible. Besides identifying certain genomic regions or genomic factors, and then you have to biologically explain or establish that as well. Okay, so these are the different research that we do in tuberculosis, looking into the pathogen from the genomics point of view, looking into the host also from the genomic point of view to better understand the disease. Just to mention, like with these genomic studies, we have been able to identify a marker for the active disease. We have been able to identify certain markers in the gene of the host which gives protection to the disease, and also people look into the expression level of the immune related genes. Not only immune, since this is an infectious disease, we primarily think that immune system is mainly responsible. Yes, it is true, but at the same time, genes with normal metabolic function has lot of contribution towards our immune function. So not only immune related genes, but normal metabolic understanding of metabolic function also leads to better understanding of the host response in case of infectious disease. With that, I would like to stop here. I would like to thank all of you. I hope I have been able to make it simple to make you understand. But yes, certain biology and the host part is more complex. And particularly if the physics students and students of other non-biology stream, if you have any problem, let me know. I can discuss now and later on also you can contact if you are interested.
So thank you, ma'am. It was really nice, and what I liked most, it was very student friendly, and I could understand also being a student of chemical science. That was a good part I enjoyed in this lecture, and it had everything. It had history, it had genomics, as well as it had the form of biology which the students can also understand. So now we can start the interactive section. I think Dr. Kuntal Bhattacharya has raised the hand. So please, Dr. Bhattacharya, can you please ask the question if you would like to?
Thank you, Nivedita madam. Firstly, I will like to thank ma'am for delivering such a nice lecture. This is one of the nicest lecture I have ever heard in my career till date. Ma'am, I have just one
Thank
question.
you.
So really nicest lecture. Ma'am, I have just one question related to diagnosis part of this TB. As we all know, there are two types of TB now, the pulmonary TB and extrapulmonary TB. In case
Yes.
of extrapulmonary and latent TB, diagnosis is getting harder day by day. So, ma'am, can you tell me what is the most authentic route of diagnosis in case of extrapulmonary TBs, like genitourinary TB or bone TB? Diagnosis is getting harder day by day. So, kindly illuminate on this, please, ma'am.
So, see, in case of infectious disease, our main aim is to identify the pathogen. That gives us like a full confidence that you have the disease. Okay, so always focus is there to identify or devise methods to identify the pathogen. So for COVID also, RT-PCR that way became, RT-PCR is for long time used in the lab. But to clinic, it came so fast, so rapidly because we want to identify the pathogen. So for extrapulmonary TB, it's difficult because for pulmonary you get the sputum which is easily
Yes, ma'am. Yes.
accessible and you look for the pathogen either microscopy and nowadays molecular techniques have been evolved where you look into the this nucleic acid meaning the DNA or presence of RNA etc. to identify the pathogen. So for extrapulmonary, so either needle biopsy or these kind of samples, if it is accessible from outside, people try to get that. Suppose like you have it in somewhere in the say other part of the body, like on the skin somewhere you see a nodule or so. So from there needle biopsy can be done. But if it is inside some other organs, like say brain, etc., then it has to be little invasive because until you get a sample from there, it's difficult to test. But one indirect way could be like nowadays even from blood, these molecular nucleic acid detection techniques are used. So if a dead mycobacterium or even a live, if it comes in circulation, then taking from blood testing is also possible. But the limiting factor is that you don't know whether it will really the live bacteria will come into the blood. And sometimes there is also a possibility of false negative, that is even if you are cured or you had long back, if there is some dead bacteria or dead nucleic acid circulating, that will give you a false positive. So actually for pulmonary TB or TB in other organs, to get the tissue is a limiting factor. So some semi-invasive or invasive techniques we have to take help of.
Okay,
So
ma'am. Ma'am,
sometimes like
I'll,
say load is not that high. Even in pulmonary TB also load is not that high. So sputum you don't get a lot of pathogens. So you have to go for like other lung fluids. So that is little invasive. You have to like stay for a few hours in the hospital, the valve fluid and so on. Okay. So the major limitation is that you have to get a tissue or a fluid from the infected site.
okay.
And then with very less amount of pathogen also because of this high sensitive molecular methods, now detection is possible.
Okay, ma'am. Ma'am, another question is, we have heard that in India or like in tropical countries, the major cause of infertility is nowadays the abdominal TB or urinary genetic TB, genital urinary TB. So,
Yes.
ma'am, in most of the cases, the patients are diagnosed during their C-section or during another type of laparoscopy, and that's why these patients are getting this disease very frequently or in a way that can be curated in future. So, can you tell me, ma'am, any ways are there for detection of these abdominal TBs or this genital urinary TB?
Yes, so if there is some nodule formation, etc., I mean, ultrasound can detect them at the preliminary level, and then further investigation can be done. And
Okay.
this urogenital, so like this, your
Or
the
abdominal TB.
yeah, or abdominal. So for this urogenital, this the pathogen can be also detected from the sample the blood coming out during the menstrual period.
Okay.
So that can be analyzed, and that is actually analyzed for if doctors suspect this kind of TB.
Ma'am, can it be detected in latent TB case as well?
Latent is a problem in the sense if it doesn't come out. See, for lung,
Yes,
this
ma'am.
latent TB remains in a uh, protective niche in a surrounded by these cells. Okay, so clinically it is very difficult to detect. We depend on some indirect test. So one of these indirect tests is IGRA called interferon gamma release assay.
Okay, ma'am.
So this assay actually takes help of the principle of memory T cells. That is like if you are infected, your T cells have the memory of that infection. And even if you are cured and later on you are infected with the same pathogen, so then those T cell expands because it has the memory of previous infection. So this principle is actually taken help to do the IGRA test. So what is done is some TB antigens. Suppose one patient is suspected, it is done from blood. Okay, so we draw blood and we stimulate that blood with TB antigens. So if the person has latent infection, then with the stimulation of these antigens, there will be rise in interferon gamma, which is actually a cytokine secreted by our immune cells when it comes or it faces any infection. So with rise of this interferon gamma, we can indirectly say that this person has TB in its system and that is reflected by the activity of this T cell by producing this cytokine. But again, this is an indirect test. So far, no very good test is available around the world for detecting latent, other than high energy NMR studies. I mean NMR instruments, but those high energy NMR instruments are not available in clinical settings. Okay, therefore
Okay.
detection of latency is really a problem. So we rely on certain indirect tests like IGRA, and besides IGRA, another older test was that tuberculosis skin test, which is done here also
Biopsy.
in the
Yes.
clinical set. So
Thank you,
if
ma'am.
you react towards that antigen that is given under your skin, people suspect that you have an infection without symptoms.
Thank you so much for
Thank you,
explaining it to
ma'am.
me.
It was really nice to answer our queries. I think it is already 12:15, and we are late in the schedule, I would say. So we can take up different questions through our feedback system.
Okay.
If we have that, and I will communicate all of the questions to your email.
Okay.
You. And it will be so nice of you if you can please answer to the same, so that we can send it to the participants.
Okay, definitely I will do it.
Yes, ma'am. And on behalf of Durgapur Government College and our whole DDC family, I would like to thank you again for giving your precious times to us. And we will like to also invite you. I have also talked to our principal sir whenever actually our institution has been taken for the elections. So we are closed enough and we are carrying out always the online classes and lectures. So we will be inviting you and we want to meet you in person actually. So we have also
Okay.
talked, and whenever our college opens, we will be glad to have you in our institution. So thank you, ma'am. So we can just end up the session here, and I will
Okay.
be communicating all of the feedback and suggestions to you, as I have also sent to Kartiki ma'am and Oringam sir. And I will be communicating all of the feedback forms to you, ma'am, also. So
Okay.
thank you, ma'am, once again.
Thank you to all of you, and I look forward to interact with you more.
Yes, ma'am, definitely, ma'am. Thank you, ma'am, for being a part of our DVD journey. Thank you, ma'am. Okay, so we are closing the session. We will be meeting again in the next Science Setu lecture, which is with Sridhar sir on 3rd May. Right? So thank you all and have a nice day, please.
Okay, bye.